Yep.
Great. Okay. Well, thank you. We should. Thank you very much. We're ready.
You can even see too. I'll just give a quick intro. Thanks everyone for coming today. So this is our series, Conversations in Global Health. And we've had a number of different speakers so far on both ends. And Alison, who is giving the talk today, is an assistant professor of nutrition. And she's been active in the Global Health Network since it started. And she's had one of our seed grants and has been very engaged. And today she's going to talk about some of her work in Ghana and Bangladesh. You guys have the slides which have changed a little bit. But do you want to do introductions, or should we kind of just jump into the talk today? What do you think?
I do want to. We can jump into the talk.
Okay. We'll let Alison go ahead. Thanks for being here.
Okay. Yeah, thanks for the invitation to talk. I've appreciated these sessions being conversations rather than formal research presentations. So at any point, I think we kind of wanted these to be discussions, and we all kind of tend back to our, traditions of presenting things and then waiting for questions at the end. But by all means, we can talk during this, and please ask questions. So Meg just gave an introduction, but again, my name is Alison Gernand, and I'm in the Department of Nutritional Sciences here on main campus. My training and background is all in public health and nutrition. I am a registered dietitian, and so kind of everything I do is centered around nutrition. Over the years, that public health focus has really been on maternal child health, and then in more recent years on pregnancy and micronutrients. So what I'd like to do kind of in the beginning of the talk is talk a little bit about pregnancy complications, and nutrition, and really make the case for these, whether we're studying them in South Asia or in Africa or wherever it is. It's meant to be more of an overview, kind of to get people's interests piqued, and maybe for people to ask questions rather than to be something to provide all the latest and greatest research in the area. So, we all know what pregnancy is, but when we're studying pregnancy and trying to think about especially how nutrition may impact it, we think about a lot of specific events that happen in pregnancy. One is the state of the uterus and how healthy that uterus is when there is a conception, and sometimes this is called uterine receptivity. We think about the implantation of that egg. Once it's fertilized, we think about placentation, which is the development of the placenta from just a few cells that start out with the fetus, that develop into this amazing organ that actually becomes part of that is part fetus and part mom. The only organ that we ever have that is actually part of two different people. Then there's a whole host of maternal changes that we often study. These include plasma volume changes. So for instance, a woman's plasma volume increases by about 50% over the course of pregnancy. And there's lots of other changes. Many women note these changes. Their kidneys start functioning at a higher rate, and thus there are more trips to the restroom. So all sorts of things are going on in the mom, actually in response to the placenta. Then you have the actual baby, the fetal growth and development, and all those things that are happening as this continuous process across pregnancy. And then you have this whole process of labor and delivery, where all these hormonal signals have to happen to trigger labor and the process of delivery. So there's lots of things when people are studying pregnancy. They may be studying at any point during pregnancy. They may be studying cross pregnancy. There are lots of factors that we try to consider, although as you can imagine, you can never consider all of them. You can never measure all of them. And a lot of these things in humans, we just simply can't really get a good glimpse at. But to get an idea of key things that often go wrong, again, so these are things that are fairly common in terms of thinking from a public health perspective. In the mom, there's preeclampsia, there's gestational diabetes. That means the diabetes developed during the pregnancy. The woman did not have diabetes before that pregnancy, and typically this resolves after the pregnancy is over. Just like preeclampsia, it's typically something that happens during the pregnancy, resolves when pregnancy is over. Hemorrhage, and of course the most extreme outcome that we want to prevent is the mom's death from complications of pregnancy. A lot of these things are difficult to measure, and we have some public health data about them, but not probably not great in low-income settings where resources are stretched and often women don't even deliver in facilities. For the baby, I would say some of the main things that we're concerned about are preterm birth, because the baby's underdeveloped, essentially at higher risk of lots of morbidities. Fetal growth restriction. Sometimes I'm going to talk about how we measure this. We're increasingly concerned about brain development and cognition. And then, of course, the most extreme being in the death of the child at whatever point that is, whether it's miscarriage. We have all sorts of ways we define this. So again, I kind of just wanted to give people this general sense, if they've seen all these terms and maybe never had them explained. People use lots of terms for poor fetal growth, and one of them, and I kind of wanted to give the sense of what we're trying to get at. So fetal growth is occurring across the pregnancy, from the beginning to the end, and really there's fairly good consensus that when we're interested in poor fetal growth, what we're really interested in is a condition which a fetus is unable to achieve its genetically determined size. So that means across the spectrum of humans, some of us are small and some of us are large. So it's not about getting everyone to the same size. It's really about everyone reaching their healthy size that their genetics allows them to reach. And the interesting thing is there can be insults in pregnancy at any of these points across pregnancy. So sometimes in general, the mom's malnourished, and other times there's an acute event like maybe an infection, or in some cases, places where there's lots of food shortages based on the seasonal changes in food availability. Sometimes it's early pregnancy or late pregnancy when all of a sudden women don't maybe have enough to eat. So we use the terms fetal growth restriction and intrauterine growth restriction somewhat interchangeably to represent this thing that we're trying to measure. And then how we actually measure it, really we measure birth weight, which is fairly easy, but in lots of these settings, there are not good scales even to weigh babies at birth. Weight changes fairly dramatically from the time a baby is born. They often lose a bit of weight and then they regain it. So the ability in research to actually measure a baby at the time of birth is quite important. And if you've never noticed this, many studies say when they report birth weight, they say, we measured birth weight within 48 hours of birth. And it sounds crazy. I see some looks on your faces like
my first daughter must have lost a pound in the first 48 hours.
Yeah, so it seems kind of crazy, but in low income settings is often how research goes, and often it's even within the first 3 days. 72 hours is kind of, they say we got to 90% of the babies within in that period. So it's kind of hard to get to babies, but that's one of the challenges in the field, I think, is being able to get these measurements, and even easy measurements like birth weight, getting them right at birth.
Is there one that's more accurate then, like in terms of if you weighed right away, and then but actually if there's a weight loss within a day? I mean, is there a period of time that's probably the most accurate to indicate low birth weight?
Well, I don't know that anyone's actually looked at those distribution. I have some data that I actually would love to get to look at this, where we had the standard measurement, like the overall trial protocol, and then we had this intensive protocol to get to every woman at birth. So we actually have weight at birth and then weight at a series of different times for the same infants. If anyone knows of any students that are good at data analysis, I want to take this on. It's like been in kind of my back pocket for a long time. But essentially, when we want birth weight, we do want weight at the time of birth. So you're essentially trying to get at how much did that baby, what's the total of the growth during pregnancy? So as far as indicators go and when you might like to measure it, as far as I know, that's beyond people have looked at. So other than having a continuous distribution of birth weight, that's really this physiologic range of measuring growth, we have ways we define pathologic growth. And low birth weight, a lot of people don't like because it's just this standard cutoff, but low birth weight is often the best we can get in some of these resources because the other measurement that we typically use here in the US and other places where we have good data on women is small for gestational age, and this requires having a good estimate of gestational age. So if you want to say they're small for gestational age, and if you'll notice, neither of these really capture what we're trying to get at is the baby small for its genetic potential. And some people do this by other measurements that I won't get into, but there are ways to add things to this. And try to get at more at if they're small, was that healthy or unhealthy? But this is typically what we do. So in settings where all we have is birth weight, we can use this cutoff. The main complication is this: if a baby's preterm, they're going to be smaller than a baby at full term. So low birth weight really, the prevalence of low birth weight is going to be preterm and small babies.
I have a quick question,
yeah.
But I don't know if there's an easy answer. So in the United States, at least, we are fascinated by birth weight. When parents send out an announcement, they're often sending the length and the weight before they send the name, right? So they'll have the sex, the age, the weight, and so we're fascinated by it. Culturally, how fascinated are other people in other settings? So we're obsessed.
Yes. I think that probably lots of us that are here could maybe comment on that, but I would say much less so. Yeah. And a lot of that's because birth weight isn't being measured, and even if it is, it's not being told to the parents necessarily. Right. You know, they're not getting some report or that cute little card you get, right, like, here's your baby's stats and here's the percentile they were at. And right. It's just non-existent. So I think that's part of it. I mean, people kind of eyeball it. Yeah. Like, they can tell if it's a very small baby or, I mean, there's this general sense of health with bigger babies, but on the other hand, there's still widespread discussions among women and practices of reducing food intake late in pregnancy because there's so little medical care. Women do not want to have a large baby.
Oh, right. Okay. So the other
aside from what you.
The other issue was the using a single cut point of 2,500 grams, whatever. I mean, that's also derived from US and European statistics, right? It's not global. The threshold value. So even things like growth charts for kids in the United States are based on all births and kids in the United States. It's not normed. It's too hard to norm to specific countries, but yet we use a threshold value that might not be the most appropriate.
Sure. Well, yeah. So sometimes people broadly use a low birth weight, even in places where we have better data, right. because we like to compare it around the world, and right. So it can be this set of comparison. But you're right. So in that way, the cut point just stays the same.
I don't mean it's meaningless. but, yeah. but on the previous slide, I think you had things like the gestational age, like the 10th percentile, for example.
You have
right?
to know which reference
you're
using.
Right, but it's the sort of thing that makes it doesn't really matter what population you're looking at. If it's the 10th percentile of that population, you kind of feel okay. Whereas a cut point necessarily conditioned on a particular weight, it seems more arbitrary.
Yeah.
That's the only.
Yeah, right.
But I know we don't have the information in every case.
Well, on the other hand, though, I can mention that there's been a huge effort to measure growth and compare it on a global scale. So it's been many years back that the World Health Organization, established the child growth standards.
Okay.
that was basically child growth from 0 to 5 years. And in fact, in the US, we've adopted that for the first 2 years of life. So essentially for children, even in the US, they use 6 different countries, and kids are measured compared to kind of the healthiest kids that we could measure from many different pockets of the world.
Right, so things are changing.
Now that's been done for pregnancy. That was called the Intergrowth 21st study, and all of that is published now. So now we have an SGA standard. We have a percentile growth reference for birth. And in fact, we have fetal growth, and some of that's starting to come out where we compare ultrasound measurements and think about fetal growth compared to. So they have 6 countries around the world for that as well. And essentially the data was such, it doesn't mean everyone grows exactly the same, but the data was similar enough that they said when they examined all of it, we can combine this into one worldwide reference. So both for kids, they had already decided that, and now for SGA. So now you'll see publications that come out now that publish SGA, they're using the new global standard. And it's really important actually for everyone to start using this so we can all have the same metric, so to speak. Whether or not we also use our in-country metric, we can do both. There's all sorts of, can't even get into it. There's all sorts of problems with how we develop these references. So for instance, they only account for the babies who are born and not the babies who are still unborn. So if you're looking at babies born at 37 weeks, there are lots of healthy babies, and we're not measuring the weight of those babies who are still in utero at 37 weeks.
Oh, right.
Right. So there's lots of, well, anyway, I think there's lots of growth we can do in thinking about this thing. So, and we're concerned about this not only for kind of immediate consequences, like some of those adverse outcomes I mentioned, but the long-term consequences, thinking about cognitive function, immune function, and then adult outcomes, and really the economic burden. If we want people to be able to use resources wisely and things like that, we really need to work on part of what I advocate for is working on nutrition during pregnancy. We can prevent a whole host of lifetime poor events that essentially mean people can't be productive and be healthy with their lives.
This is one of the, so the dollar amount just seems extraordinarily low to me. Is that because
it does seem low, but that's for low birth weight. So so it's not necessarily for all kids who are unhealthy. And it's been a long time
so
since.
You could be low birth weight but healthy.
Yep.
And so you're effectively zero.
It's a low birth weight, but this estimate. I haven't looked back at this paper in a while, so unfortunately I can't recall exactly what they did, but I believe it was in low-income countries.
That was my question.
Yeah, so I don't think it actually included any data from high-income countries. The numbers on low birth weight are a little bit low, but low. They're a little bit old. But you can see some regional differences. For instance, in Asia, South Central Asia, where Bangladesh is, the rates are quite high. A lot of this is due to growth restriction and not just preterm birth. But this problem, as well as preterm birth, they have not been going away. In fact, preterm birth in the US and other places has been increasing. And in Western Africa and Ghana, it has some of the higher rates of growth restriction. So that first column is just low birth weight, and then the second column is their estimated IUGR, growth restricted low birth weight. So you can see how those numbers differ, and they typically drop off, but you can still kind of see where some of the issues are. Can they see the slides? Are they?
They have them printed out.
We actually can see the slides too.
Oh, great. Okay.
Yeah, I think we have them printed and we can see them.
So then I'd like to highlight preeclampsia and then preterm birth. Preeclampsia is a really complicated condition that people that studying it just really been trying to describe what it even is. Do we call it a disease? Do we call it a syndrome? So you'll see all sorts of names for it. We've typically defined it by newly onset hypertension. So the woman, kind of like gestational diabetes, the woman's developing that during pregnancy. And then this presence of protein in the urine. But now there's kind of a more complicated definition, a much more complicated definition. Its prevalence is fairly similar across the world, both in low and high income countries. Perinatal mortality is 5 times. So we're concerned about it, not just because it's kind of puts the mom at risk, but also there's risk for the baby. So perinatal mortality is 5 times as high. And often the only cure when the woman's condition is becoming extreme is for the woman to deliver the baby. So it accounts for, there's an estimate that accounts for 15% of preterm births. So essentially, the clinicians have to decide to deliver the baby. And it's indicated in up to 1/4 of maternal deaths. So people are really concerned about it because of those adverse effects, not necessarily for it itself.
If people are hypertensive before they're pregnant, that's not considered preeclamptic?
So there are new categories now. So there's something now, so like I said, it's getting more complicated. So there's something called superimposed preeclampsia. And that means you had hypertension before you were pregnant. Mm-hmm. And then when you went into pregnancy, at some point you developed these other characteristics that are like preeclampsia. But before they excluded those. There's also more mild classifications now, including gestational hypertension. So you didn't have it before, but you developed hypertension. So there's kind of these tiered conditions. So accounting for that, like whether or not you had blood pressure before pregnancy to know if the woman developed preeclampsia, right, is then an issue of, was the woman even going to medical, was the woman getting any medical care before pregnancy? Which is part of what's driving my interest in studying pre-pregnancy. So we can get some of this information ahead of pregnancy. So preterm birth continues to be one of the most important adverse outcomes and really has a high prevalence, unfortunately, in the US that's similar to the prevalence in most low-income countries. And it's just it's a huge cause of infant death for a variety of reasons. And preterm infants, I think a lot of this, people understand, but there's, in this country, infants can survive at much, much younger ages, but in other places, we just don't have the medical expertise or technology in place to be able to save those babies that are born early. As well, it's harder for the mom. So there's lots of things, talking about these changes in the mom. We think about like wanting everyone around the world to breastfeed. Well, when babies are born preterm, the mom's baby is less ready to provide for that infant ex uterus. So there's lots of things as well. It's harder for that baby to breastfeed when it's tiny and things are underdeveloped. So we want things to go even better, even if we try to have these better interventions like breastfeeding. Still, preterm birth often prevents a lot of that from happening. I included this chart on preterm birth and stillbirth just to show that there's lots of different categories, and it seems like the etiology of some of these conditions is different, whether or not a baby's born very preterm or what they call moderate or late preterm, as well as when people are classifying what they call stillbirth, what they call miscarriage, things like that. So when you read these terms or read prevalence, if we're going to compare them around the world, we really need to know how people are defining them. And as well, we need gestational age. We need an accurate measure of gestational age, which is just often difficult to get. Okay, so I can't give a talk, I feel like, these days without talking about the placenta. It's likely become my favorite topic. And its importance cannot be underestimated, but it's often overlooked. So the places I've been able to work in a few of these settings, Bangladesh and Ghana in particular, when I ask people about the placentas, people essentially don't look at them. They all get tossed. There's just, I don't think it's that people aren't interested. I think it's just that people don't know enough about the placenta, or that they should be looking at the placenta. And you know, honestly, that even happens in the US. I mean, we send only kind of the placentas of the pregnancies where there was some high risk or concern to the pathology lab. So we don't really even have that much information on normal placentas because we don't study them. So we think of placentas, I say we, I feel like there's this general sense that people know the mom has nutrients and oxygen, it needs to get to the baby, okay? So the placenta is the thing that it goes through to get there. But really, the placenta is controlling what gets through, how much, and it's metabolizing things. So it's breaking down and rebuilding, nutrition for the fetus. It's also making hormones, and it's controlling essentially everything about what's happening in pregnancy. So it's sending hormones to the baby, and it's sending hormones to the mom, and making all of these things happen. If there was no placenta, then the mom would not know she was pregnant, and all those things wouldn't happen. One of the things that's important about the placenta growing is the blood flow to the placenta, and then I also just want to point out the placenta grows ahead of the baby. So it has to do all these things for the baby. So essentially it needs to grow fast first. It keeps growing across the whole pregnancy. But the majority of the growth is completed before the end of the kind of the third trimester, when the baby really has to kind of have this explosive growth. And not to get too detailed, but I just wanted to point out, I'm not sure if I can actually point to things, if they can see this in Hershey, but that this is kind of a cross section of the placenta. And this part, all these vessels, this is the baby, this is the fetal cells. I mean, it's the placenta, but the placenta that developed from those original fetal cells. And this is the mom. This is the part of the uterus that the endometrium that became part of the placenta. And these are the mom's blood vessels. And essentially what happens is these blood vessels are almost like a water hose, and it doesn't end in some capillary system. It just ends. It's an opening, and it basically gushes maternal blood into this space where then there's a thin membrane here that's fetal cells that where the mom's blood doesn't actually touch the fetal cells, and that's where all this activity happens. This part of the tissue metabolizes things, makes hormones, and as you can see, if it makes hormones, it can release them to the mom, or it can release them to go down here that goes down to the baby. So this is a cross section of what gets delivered as the placenta, and it literally has mom cells and baby cells that have grown together to make this really amazing organ. So part of the reason I pitched again for it is that I think we need to be studying placentas around the world in all these different settings, and we don't know a lot about placentas, and we especially don't know a lot about placentas in low-income countries. There's lots of different diseases and pathologies we can look at, and I won't mention much about these other than sometimes the way the cells invade, the fetal cells invade, causes problems, whether it's adhered, it's too invasive or not adhering well enough. It can get infected. You can develop lesions. The placenta can grow too much or too little. We often compare its weight to the fetus. And then the cord can be, the umbilical cord can be inserted on the margins, and that can cause problems with blood flow and transfer of nutrients and things. Okay, so that's kind of pregnancy aside. Hopefully you can think about underlying nutrition issues, but there are lots of connections between maternal nutrition and what ultimately happens in terms of the health of that baby. But But one kind of theory and process that I've been looking at myself and has been around for a number of decades is this idea that the mom's nutritional status impacts that expansion of her plasma volume, which can cause inadequate increase in her cardiac output, which then decreases that blood flow that we need to the placenta for all those nutrients and things actually to get to the placenta and ultimately to the fetus. And that reduced blood flow can either decrease the placental size, so the placenta is not growing well, or it can reduce that and nutrient transfer, which results in poor growth of the fetus. This is just one of the pathways that's proposed between mom's malnutrition and poor growth of the baby. There are many other potential pathways. So I'm most interested in micronutrients. And I'm interested in how the mom's micronutrient status impacts the pregnancy, the progression of it, kind of all these things we've been talking about in the health of the baby. But I'm also interested in kind of what's the prevalence of deficiencies around the world. So we call them micronutrients because one, they're nutrients, they're these essential vitamins and minerals, and they're things we get in very, very tiny amounts in our diet. So other things like fats and proteins and carbohydrates we get like in very large amounts, like that's the bulk of what we're eating. But there's these, you know, iron, zinc, vitamin A, all these things, they're in such tiny amounts in our food, but they're completely essential to our health and the function of our body, and they're essential to things that happen in pregnancy. So the highest deficiencies around the globe when we're kind of assessing things globally are iron, zinc, vitamin A, and iodine. And I've kind of added vitamin D. It's kind of been emerging as a bigger health issue, even in places that are tropical, that have a lot of sunshine, and people should be making a lot in their skin. So how do we measure micronutrients? Well, so, you know, there are things that we eat for the most part. So vitamin D we can also make in our skin. And so a lot of people measure what people are eating. They measure dietary intake. But really this is a proxy for your status, because so many things happen between what you eat and even how the food's prepared and what actually ends up in the food that you eat. Between that process, even if you're measuring the food that someone is consuming, and then this process of it going through digestion, being absorbed, being metabolized in your body, things like that. So we can measure diet. That's a bit somewhat easier to measure, but we measure that really as a proxy. And then we have a variety of biomarkers, and typically these are in blood, measuring in serum or urine, serum or plasma, but sometimes in urine as well. There are a few other ways to measure micronutrient status. Some people take hair samples, like fingernail, toenail samples, and some of that's to be less invasive rather than to pick the best method. But for the most part, I've always used blood samples and measuring concentration of those things floating around in your blood, retinol or iron or zinc. So here's an example. This isn't my work, but it's an example of where someone actually measured multiple micronutrients at the same time. Typically, it's pretty expensive to do, and people typically measure one or two things at a time. And some of these won't have familiar names, but you can see in this population, zinc deficiency is over 60%. This is in a population of women in early pregnancy, and this is in a rural area in Nepal. This is an indicator for vitamin K deficiency, and you know, surprisingly, it gets little attention, but in this population, it was 40% of the women were vitamin K deficient. B6. So some of this is to point out in every population, they're kind of unique deficiencies. And so measuring them and measuring many of them to really get a glimpse of what the problems are is important, but it's challenging and costly. And here's a map of vitamin D deficiency. This orange is this orange color is the getting to be the insufficient range, and the red color is deficiency. And most of this gray area is just places where we don't have good data to even say something about vitamin D status. So you'll see most of Africa, we have very little data, but in South Africa, we do know that there is deficiency. And then interestingly, as I said, even in places that should be getting adequate sunshine, like India, there are high rates of deficiency. Other places where it's a little better, we fortify our foods, like in the US, we fortify things with vitamin D. So even context, studying each micronutrient, it really depends on there's a lot of factors to consider. I'm definitely not going to talk about all of this, but in a recent review paper, we were trying to connect what we know about micronutrients in each of these different stages in pregnancy. So I mentioned there's all these different events. There's the maternal status, preconception, there's implantation, there's what's going on with the placenta in terms of those blood vessels developing, the vascular network, and nutrient transfer. So for each of these cases, you'll see there's a variety of micronutrients. The vitamins are in blue and the minerals are in this red, pinkish color. And so micronutrients are really important across the pregnancy. There's probably a lot we don't know to truly fill in this chart, but we do know a lot about what micronutrients are needed at different stages of pregnancy. There's also increasing work on the role of micronutrients preconceptionally. And so I won't highlight all of these things, but if you'd like to look at this paper, this was kind of a good summary of the role of micronutrients before pregnancy occurs. And they cover a wide range of micronutrients. And again, that's been my increasing interest. And I have a personal focus on vitamin D, so I just wanted to highlight some of the complexities. Vitamin D has unique complexities because we make it in our skin, but all of these micronutrients kind of have complexities to them as far as how we study them. So in the case of vitamin D, we can eat vitamin D through very few foods, but fish has vitamin D. We can also make it in our skin, and this gets converted to this metabolite called 25-hydroxyvitamin D. Well, this metabolite is not what our body uses as vitamin D. It's not. It doesn't have an active role. It just circulates around. So the mom this has to go to the mom's kidneys to get converted into this, what I've abbreviated as 125 dihydroxyvitamin D. That's what our body uses. So there's kind of this process from this parent vitamin D down to what we mean when we say the effective vitamin D. And in fact, that particular metabolite doesn't cross the placenta. So the placenta is transferring, and I told you it has this active role, it's transferring this intermediate to the baby. And the baby eventually has kidneys and is making its own biologically active vitamin D. So the placenta, though, is a metabolically active organ, is making it itself. And that's some of where our knowledge ends. We don't know a whole lot about what the placenta is actually doing with this, although we do know a little bit about its importance in developing vascular networks. So for each of these micronutrients and kind of getting a glimpse of them and how we think about them in pregnancy, is a bit complicated. I think I won't say too much about this, but I did want to point out there is a standard UNICEF multiple micronutrient supplement formulation. So I've listed here the RDAs in the US for pregnancy. And for the most part, this supplement that's has been tested a lot worldwide is not in recommendation everywhere yet, but it's pretty similar to what our RDAs are here. So there's been growing evidence that giving multiple micronutrients during pregnancy can prevent preterm birth and fetal growth restriction. So just for anyone that might be interested in collaborating or interested in nutrition research, I just wanted to mention some of the ways that that I examine this topic of micronutrients in pregnancy. So I use observational studies or have conducted studies observationally. So the new work that I've just started in Ghana is enrolling women actually before pregnancy, and we're going to try to examine micronutrient status across the pregnancy. I've also worked with randomized controlled trials, giving either multiple micronutrient supplements or vitamin D supplements, and the vitamin D supplementation, both of those trials have been in Bangladesh, one in a rural area and one in an urban area. And then since I've been at Penn State, I've been able to work with other researchers here using mouse models to study some of these deficiencies. So things we can't do during to humans during pregnancy, to look at placentas in the middle of development, we can do in mice, even though there are some differences. Interestingly, their placentas are about the closest to humans that we can get. There's lots of different types of placentas, and interestingly, mouse placentas are fairly similar in the grand scheme of mammals. So then let me just highlight some of the trials. Again, I didn't see this as a research talk to talk about findings, but I thought I'd highlight kind of just some of the methods and field challenges. So so when I was at Johns Hopkins, I got to participate in this trial that was named the JAVITA. The original trial was named JAVITA because it was a vitamin A trial, but anyway, nobody cares about the name, but it looks a little odd. So this is the third trial they conducted in the same area, and it was a very large randomized controlled trial that enrolled almost 45,000 women. So if you imagine, you can't measure micronutrient status in all those women, you know the effect of the micronutrients you're giving, so you can kind of study these bigger things. You can't actually measure micronutrient status in everyone. So within this two-arm trial that enrolled women, it was actually tracking women from before they were pregnant, but they started getting supplements in pregnancy. I was able to nest this intensive pregnancy study, and we studied we enrolled 500 women. We set up an extensive labor tracking and delivery system so that we could go to the women's home and be there when they delivered. And if anyone knows Bangladesh, this is the area, this is the area where the study was conducted up here in the Northwest. This is an agricultural area. Due to irrigation advances, there are 3 growing seasons a year. There's water everywhere, and they eat a lot of rice, lentils, vegetables, and a lot of fish. The literacy rate is fairly low, and the malnutrition rate is very high. There were, I believe, 5 women in the whole study that were overweight, and that was like by only a little bit, but around 40% of the women were underweight according to BMI, and almost everyone delivered in their homes, and about half of babies in this area were low birth weight. So there's a lot of malnutrition, and it's a setting where we're very interested in because we want to help figure out what can be done to prevent these poor outcomes. So this is just a glimpse of what we did. Women were enrolled fairly early in pregnancy. We got height and weight and body composition measurements, and I'll show you a picture. We actually set up little kind of mini clinics to do ultrasound work, so we'd actually have an accurate measure of gestational age. Then we kept doing these same measurements during pregnancy, and then at birth, we actually had a system where we could show up and weigh their placentas, get birth weight at birth, and collect cord blood. And this is just to give a sense of when we're collecting this blood, we collect mom's blood and cord blood, but again, the placenta is making all these things. So when I want to define what these, some of these are growth factors and some of these are related to that vascular development, angiogenesis. And so some of these things might be coming from the mom or the placenta. Some of these things may be coming from the fetus that we're measuring in the cord blood. So it's kind of a lot to think about. Even though we get the samples we want, we're not even always sure exactly, depending on where it shows up, where it's coming from. And then here's a few pictures of the measurements. We did these measurements in the home. We needed to go to these moms. It was a large study area. Again, we got body composition and things like height and weight. This is what our little mini ultrasound clinic looked like. And this is one of the positions. We had a portable ultrasound, a portable Sonosite ultrasound machine, which is quite durable and field friendly. You can drop the transducer and it will not break. So it's a really amazing machine. I recommend it highly to anyone trying to do ultrasound work out in a field setting, especially where you need to move it around. We had to train a team and basically have them be on call all the time for these labors. We kind of had to turn the Land Cruiser into kind of a mini ambulance. So that if there was a complication, we packed everyone in the car and went immediately to the nearest medical facility, which typically from a drive, if it wasn't the rainy season and roads flooded out, it was less than an hour. But you can imagine for things like cord prolapse or hand prolapse, you know, there were some moments where people were pretty nervous about getting these women to care. And then, as I said, we did all these measurements in the home. So these are the data collectors dealing with the placenta. We had to come up with all these materials, you know, drawing cord blood. This was a floor. This is the floor of the home. The mother is over here, which I don't show. She's just delivered on the floor of her home. And so we just set everything up here. And here's us weighing the placenta, weighing the baby. And this is what this room looked like from the outside. Okay, so I will just say a few more things so I leave a few more minutes for questions. The current trial we're conducting is a vitamin D trial. There's a lot of vitamin D deficiency in Bangladesh. This trial has enrolled 1,300 pregnant women in mid-pregnancy, essentially because that's when they show up. But in this case, we're interested in the vitamin D that's really transferred to the baby, that the baby is born with high vitamin D status. But I'm looking at pregnancy events. I'm looking at the development of the placenta and what's going on with the placenta. So a lot of that work is in progress. These women, however, are delivering in a government hospital facility. Most of the women in Dhaka and in an urban area are not delivering in the home. So here's just some pictures of field work. You know, we always have to set up places to do blood measurement and just do the best we have. We had a very tiny area. This picture, I guess, is kind of turned a little weird, but we had a very tiny area to measure placentas. So the technicians really didn't have much space to draw blood and do these placenta measurements. In this study, we're actually taking placental tissue and bringing it back, sending it here. And then our current work in Ghana, we're enrolling women pre-pregnancy. Our ultimate goal is to follow them across pregnancy. But for now, so I don't know if those in Hershey can see this, but the star is covered up here. But we're working in an area that's just a few hours from the capital that's right on the edge of Lake Volta. So you can't see it behind this picture. But it's kind of a semi-urban. And we started this pilot work initially before we did a full study just to find out if women would even tell us they expect to become pregnant. Because here often there are many relationships that are essentially marriages, but they're not formal marriages in the way we think about them in the US. Typically, no one's taking blood samples, and everyone told us like no one's going to want to give you their blood. So far, we've gone check and check on these two things because women will tell us. They've been quite free with that information, and they've allowed us to take blood samples. So now the main question is, can we track them and follow them until they become pregnant? So those are our main challenges. We're hoping the third challenge will also be okay. So I won't talk about broader implications. I'll put this slide up because I've worked with lots of great people, and I do want to acknowledge my time at Penn State has been supported by the Birch Award, which is managed out of Hershey, but it's for all of Penn State. There's faculty at both medical campus and here that are part of that, and that's really given me the support and time to do a lot of this work. Started with it so I can lead to other things.
Great, thank you. It's really interesting. I have so many questions, but I'll start with Hershey. Do you guys have any questions to start with?
I do. Can you hear me?
Yes.
Yes.
Okay. Hi, Allison. This is Carol.
Yep.
So my question has to do with pregnancy history in these women. How much do we know about how a history of repeated pregnancies might either deplete micronutrients in the woman, or affect what's happening in the placenta, or could it have the opposite effect because a woman might build capacity to have a healthier pregnancy over time? Do we know anything about that?
We know some about it. So we know enough that there are recommendations that say women should space their pregnancies by at least 1.5 years, preferably 2 years. Now, some of that's not necessarily based on just micronutrients. It might be based on, overall health indicators like, BMI and whatnot. But that's kind of a general recommendation for the very reason you bring up, to allow the woman's body to kind of recover nutritionally. And I think some of the reason that a woman with a healthy pregnancy has other healthy pregnancies is actually more selection bias, if I should call it that, because the women who are kind of healthy and have healthy reproductive system are more likely to have, healthy pregnancies. So I'm not sure, I could be totally wrong on this, but I'm not sure I've ever seen anything that says because you've had a baby, it means you're more, it'll be easier to have a second healthy baby. However, on the flip side, women that develop preeclampsia during pregnancy often go on to have other complications. And so people aren't just concerned about those immediate things now. Now we're kind of concerned about long-term, what happens to the women.
Great. Anyone else on your side?
I had a question too, kind of. I was wondering, I don't know if anything's been done on this, or maybe this isn't. My question is, with across the globe, are these thresholds for deficiencies, should they be consistent? I imagine that it's the same number threshold for vitamin D deficiency in every country, but is that something that could be environmentally, the environment could impact it, and maybe in India they don't really need they don't need the same threshold of vitamin D as we would in the US. Is that possible, or has that been looked at? What do you think?
Yeah, it has been looked at. There's actually, you've hit on like one of the biggest controversies in micronutrients. Right now, a lot of other places use our thresholds that come from the Institute of Medicine, that we set for deficiencies, and they don't necessarily have the data to set their own. But then there's all sorts of complexities, like vitamin D, the deficiency is set based on bone health outcomes only. And so any of these other outcomes it may be related to, really, we don't know kind of what the cut point is to help you be less at risk for that. So there's actually a lot of complications in thinking about deficiencies, and that's the reason that we like to measure either a blood measurement that's continuous, or we like to look at data in terms of not just how many people are deficient, but the range of concentration, so we know what particular levels are associated with what particular outcomes in that setting. So typically that's how people should do the research, rather than just using some predefined cut point.
Okay, thanks.
Great. Does anyone on this side have any other questions or comments?
You mentioned that the vitamin D status in India and Bangladesh are pretty low. Do you know why, reasons why that is?
We don't. I've been working on some new data analysis to look at this. And, you know, people use these regression models and they say, oh yeah, there's all these factors related. And it's all the stuff we already know is related to vitamin D. They're not getting much in their diet, and they're not, you know, they're not spending enough time outside. They're wearing too many clothes. So we're trying to figure out kind of what are the main driving factors, especially people used to not be getting vitamin D. So I don't really have a great answer for you now. Some people think that some of it could be pollution. So the sun isn't, you know, the UVB rays aren't actually reaching us, but a lot of it is because people all around the world are spending more time inside. So it's not just us in our office jobs, but it's like people in factory jobs, or just in settings where they're not, you know, they're not typically out in fields anymore working, or doing things where they're walking as much, or just being outside as much as people used to. So there's lots of people trying to figure that out. We don't really have a good answer yet. It's kind of unfortunate that we need to figure it out, but.
So have you talked to Nina Jablonski in anthropology?
Yeah.
One of the world's experts on this.
Yeah, right. So here at Penn State,
Yes.
we have kind of a world expert on skin, and I know she's been trying to measure lots of different factors in her work in South Africa,
Right.
thinking about pollution and.
But she's also getting, in the map you had of the world where it was mostly grey because there's no data, but they're trying to use some of the climate and weather information, sun
Yeah.
exposure, to actually approximate conditions in all parts. I mean, it doesn't matter, right? You can see the whole
Right.
world.
Right.
So
right.
they're looking at it from the other side of it to you, perhaps, but it's helpful.
Right, well, it's about 2 o'clock, so I'm gonna wrap things up and just say thank you